Okay, sorry about it. So, by the way, this is a picture where we actually store data in bacteria and the DNA, and we demonstrated to the Prime Minister of India and Israel in 2018. So, that picture we are showing here. So, this course is for an emerging area because I think right now you are more active on the web, right? With so many things, for example, taking pictures or putting it on Facebook or doing a lot of Twitter, everything, right? All kind of videos you are posting. Uh, just one tea time problem from the beginning. What is the size of YouTube? Any idea? So try to find out by doing Google search or try to estimate the size of YouTube. It's really interesting, you will see that it's huge actually. Because every minute how much content is going on the YouTube. So the question is, for these storage, you require a lot of cloud centers these days, right? So you store data in, so you must have heard that Facebook has made data center in the ocean somewhere, or Google has made all kind of things because it requires electricity, all other kind of things. So it's a really big burning problem of data storage. So this course is all about data storage, but new emerging area, although this basic idea was there in 1960, but the main result was obtained in 2012. Okay, so that is something we were also doing the similar thing at that point of time. Actually, it is, I still remember in the summer of 2012 here. So this idea of DNA storage was experimentally showed by George Church at Harvard. And since then, immediately after that in 2013, Nick Goldman and his team, Nick Goldman was at Cambridge at that point of time. So they did another experiment by storing data on the DNA and retrieved it successfully. We also at the same time, so I remember I asked one of my students, Shalin Shah, a BTech student at your place, and I asked him to look at the Nick Goldman paper and implement it, right? And then we realized that there is a problem in the algorithm, so it can only encode up to 500 MB data. So we tweaked the algorithm, and that resulted in the software DNA cloud. And since then, we have done some of the other work. And so that is interesting. This is how we involved. So I think almost at the same time, when George Church and Nick Goldman, they did the work. So I'll show you today some administrative part of the course and some historical thing, how the basic idea came up. And then now there are a couple of approaches. There are a couple of companies, in fact, in Silicon Valley, there is a DNA Storage Alliance under the ISNIA. ISNIA is a standard organization, non-profit organization, which handles a lot of companies and who are working in DNA, who are working in storage actually. So DNA storage is one part of it, okay? And so almost all companies like IBM, Microsoft. Microsoft actually started the first paper in 2016. And since then, they have, I think, written about 35 papers. Many of them are very good in nature. In fact, Shalin, after that, he spent some time at Microsoft Research after here, and he has done some work there also. So it's a really interesting thing. So you will see that different aspects, what are the areas, what are the challenges, and how companies are dealing with it, right? So overall, in the whole course, you are going to learn all these different things. So by the way, I think sometime I show this is a first program written by bacteria. Hello world, okay? So you can see that wonders are possible when you mix computer science and biology. So now I'm going to talk about administrative details of IT 495. By the way, all this is part of another branch known as natural computing. So there are a lot of books available right now, unconventional computing. computation in living cell, different kind of computational aspects. I think if you want to learn more about different aspects of natural computing, you can look at the course, which is also available on YouTube. You can actually watch previous course. Okay. So this course is only on one segment, which is DNA storage. This anyway, So I think first exam is somewhere next slide, right? Yes, 11th September to 14th September. Although I thought that we should not take any exam, but some few things you can ask. Only 4 questions, right? So should not be difficult either, right? And so I would like to involve, because the field is emerging and every day new things are happening. So I'll show you some news headlines, what is happening every day. Even something happened recently on 5th July. Okay, something happened today also. I received something in my mailbox. So basically I'm saying that this is evolving very fast. Okay, so now it is difficult to even look at all the literature at one point. So maybe you can also look at the literature, organize it, and it's a very interesting thing. So you will really be at the top of the new area. Okay, so timings, right? 10, 11, 11. Yeah, so the grading policy is something 30%, 20%, 50%. I have kept it to project. So obviously, project has more component. And remember, it's always tentative, because I may optimize it towards the end, depending upon the response from you, right? Slightly. If you're doing good here, I mean, in fact, I will encourage that you should focus here more. And this time, since many times when you take project, you are lost because there's so much information and you don't know where to start, what to look at. I would like to work with you. So probably I'm going to send an email pretty soon so that we can form for the specific topics based on your interest. Somebody may be doing something, somebody may be doing something, right? Depending on something meaningful can come out of this whole class, right? So that is the target. And obviously Diwali break is there. Holidays, you should know. Attendance policy, I mean, type 2 questions, T-type problems. Okay, so what kind of opportunities available? You can do, see, everybody has different aim. Somebody wants to go for jobs in good companies. By the way, all these good companies like Microsoft, IBM, they are all hiring in DNA storage. Okay, Intel, they're all working on it. So they have this division now. Seagate, okay, Seagate is another company for storage, works on the hard disk. All these companies, around 41 companies, they formed a, so there are available job opportunities as well to work in this niche area, because everybody wants to push the technology ahead and take the lead. Who is going to create the first affordable system? And software development is, of course, is a part of it, right? I mean, so there are different aspects depending upon your shraddha, right? So if you have more shraddha towards developing something, so you are welcome. So I can give you those kind of things. We are also, so as I mentioned, we have done the experiment. We have stored data on bacteria and retrieved it successfully. So we have a lot of interaction with experimentalists, So if you have really something and we want to do an experiment, so that is a possibility. Okay, you can actually do an experiment. We have that kind of collaboration available. Okay, so now that was administrative part. Now we are going to talk about elephants, right? How to store elephants. So elephants is a metaphor, right? So we used, in fact, is it possible to store elephants on gut bacteria? That is a possibility now. Okay, so how? What is this elephant? So first of all, I'm going to give you some idea about storage. So storage is the fundamental need for both life and computation, right? Have all your books stored in your bag, It's a very primitive need, right? And every life needs storage of resources such as food. So also basic computing primitive. And many times it is said that life is nothing but storage plus information processing, that is computing. That gives you different kind of life forms on the planet. And we as a modern human stores data. So that is these are some examples of natural storage. I don't know whether you saw this. This is a honeypot ants. So they store honey in their stomach, right? So in the nature, you will observe different kind of storage mechanisms. Harvester ants are there, so they are storing their food, right? Here is another bird storing another one. It's a very common phenomena, right, among the different species that they want to store something, right? Especially, I mean, they are focusing on storage, but we are worrying about data. Okay, so that is a key difference. So now I'm going to give you a brief idea about, it's a very brief summary of, so story of data storage. Okay, so some of you might be, I think your age is something around 17, 18, maybe 19, I guess. So before 19 years, you might have seen. So anybody has floppy disks at their home? Do you know what is floppy? Okay. So some people might have seen those. We have used those things, where the size was only few MBs. So those kind of things are there. So I'll show you some of the pictures here now. So cave paintings. This is the first primitive storage thing, right? See? So you store information on the caves, walls of the cave. That is also data, right? You are storing data on the caves. That's pretty old actually. Tally sticks. This is also a very primitive thing, around 5000 BC, So even they used to, store on the bones, they used to make marks and something like that. So tally sticks to store different kind of things. Papyrus was early form of paper actually. Okay, so this is also around 300 BC, before 300 BC. Paper came around 100 BC, and as you know, it was discovered in China. All this storing data. Punch card was a very interesting thing. I think as a computer science student, you must be knowing that the first computer program was written in punch cards for operating looms, in Europe. So this is something very interesting. And I think I don't have that picture right now, but you can find the first picture. It's called kind of a painting created on a cloth using punch card system. So you have to write a computer program on punch cards. That's another interesting thing. Okay. Phonograph. So phonograph was developed by Thomas Alva Edison. As you know that. And actually, that was really interesting thing. Actually, how you can store. I think, so I don't know whether you visited the Edison lab in New Jersey. Luckily, I have visited once. It's a very interesting lab, the kind of stuff that is lying there. And you can see the very, very early stage, It's a very small lab. I think maybe this much size, right? And all discoveries he did there. After that, he became successful businessman, right? When he found General Electrics and so on, right? So it's a very interesting thing when he was doing experiments. Okay. There is a telegraphone. Okay. So all these patent things started actually in 1800, at that point of time. So magnetic tape was discovered by this German engineer, okay, in 1928. Williams tube. So first random access computer memory, actually. So you see, these are very big things at that point of time, right? First hard disk in 1956, the IBM 350. You can see that how huge the hard disk is, right, at that point of time in 1956, right? And it was only able to store just under 5 MB. This much size machine only storing 5 MB. Will you buy it now if it is available? Of course not, right? Because you see the evolution, how things have happened, right? Really, things have changed. In fact, another hard disk with capacity more than 1 GB in 1980. So this is 250 MB hard disk, left side, 79. And in 1980, IBM 3380, the first gigabyte capacity hard disk drive. I think the cost was something around $81,000 to $140,000 and 250 kg weight. So the similar things are happening for DNA right now. Okay, so soon you will see that a pen drive kind of thing you insert and do all the jobs, right? So you are in the same age. DRAM in 1966. I think some of you might be knowing about DRAM, right? Then 8-inch floppy disk, 71. Okay, so storage size is around 100 MB. 5.25-inch floppy disk in 1976. 3.5 in 1981, which is 1440. I mean, so ranges from 360 to 1440 kilobyte. Different floppy disks, actually, you can see. I have some of these things in my home yet, So my daughter asked, what are these things? So I say that these are the old storage device, but they are lying anyway. Compared this, I think CD you might be knowing, right? I think by the time probably CD you know. What is the size of CD? I mean, how much data can be stored on CD? 700, correct. So I think normally when I was studying in those days, 90s, so they used to release CD for different softwares. So for example, Linux TrackViewer CD, I remember, you insert in the computer and try to install those things. It's a very interesting phase. CD-ROM, then MOD, magneto-optical disc in 1990, in different capacities. So actually, all this development happened after the formation of ISNIA. So ISNIA, as I mentioned, is a standard body of different companies and academicians who actually develop standards for storage. Okay. So compact flash was in 1994. Some of you might be using the camera. I don't know whether you still have that camera with you or not. DVD movies, so dekhi honge, right? I mean, this is something very common, I guess. Memory stick by Sony. One company, Philips Research, has done a lot of work on storage actually, and I will show you one of the person who has done a lot of work there. By the way, USB flash drive is really remarkable thing that appeared in 2001, right? So now you can carry data on your small disk, right? Pen drive. This is something really remarkable. Blu-ray disc, launched in 2003. HD DVD, high density, in 2004. So capacity around 15 GB to 30 GB and so on, up to 51 GB. And then, so these are the different developments. This is a cloud data center, IBM Cloud Data Center in Auckland, and this is in South Korea, right? So a lot of stacks of disk, connected in, while storing data. This is Google file system. I think you know that in cloud, how the data is stored. You take the file, divide into chunks, and replicate chunks in different servers in the cloud, right? And when you want to recover the file, so there is a master server which knows where which chunk is. Okay, and there is a replication of master server also, because if master server fails, you are gone. If one master server fails, next master server will take care of everything, right? This is how the whole system is designed. So it's really very interesting thing. So this is summary of whatever I have shown you, from cave paintings to cloud storage that you use every day whenever you're using your Gmail, right? Or whatever, or Facebook. So everything is through cloud. In the cloud storage also, a new thing has come up in terms of codec, which is known as regenerating codes, which can actually optimize data download in case of repair bandwidth. I mean, so that is repair bandwidth when you're repairing a node or different kind of things. It's a very interesting thing. It started in 2007, but anyway, I'm not going. Microsoft Avalanche file system is based on that. By the way, this is a comparison. I mean, this is a tweet by this user. 1958 and 2021. Can you identify this? This is a early IBM computer, and this is a IBM quantum computer. See? So the same thing is happening for even quantum computer at a similar age, right? 58, 50s, or towards a 60s, and I mean towards a 58, 59, 60s. that comparison you can see. By the way, there are some heroes here. He's Imig. He's very well known, more than 1100 patents. Most of the time he is spent in Philips research. And he has all patents on digital audio, video, CD, DVD, Blu-ray disc, any kind of storage, you will find his patent. Very remarkable person. I call him father of storage technology. We are in touch. I will tell you, he gave some problem for us when he read our paper. So I'll share that problem with you. And Samuel Edison, he invented, he was one of the inventor of pen drive that he used. Okay. So because of these two people, you have seen remarkable progress in the storage. So this was the early history. Now we were talking about elephants. I would like to give you some idea how much burning problem is this storage. It's really very, very serious challenge. So you will see pretty soon. Although some of the comparisons are old, but so elephants mean big data, right? So much data that we are generating. So there is a estimate by Eric Schmidt, executive chairman Google, in August 2010. This is very, to give you some idea. So how many elephants are we generating? From the dawn of civilization until 2003, humankind generated 5 exabytes of data. You might have heard of kilobyte and megabyte, gigabyte, exabytes in the order of 10 raised to the power 18. Okay, 10 raised to the power 18. So 1 exabyte is equal to 1 billion gigabytes. And now we produce 5 exabytes every 2 days, and the pace is accelerating. Any idea today how much data we are generating every day? Yeah, so actually it is estimated by 2025, we will be generating 463 exabytes per day. Now you see the burning issue. So much data, you don't realize, immediately you take a picture, post it, right? So it's a problem of the headache of the companies to produce the services, right? It's a really challenge as an engineer, right? So this is something for the entire humankind, they generated 5 exabytes of data, and then, okay. So this is the estimate. By the way, we have also done some estimate. What is the size of internet? The size of internet, something around 700 exabytes. We computed some few years back. And so how to store this much? Of course, it is in distributed manner, right? If you want to store data, what is a basically very good ultimate storage device that is possible, right? Soon, because of Internet of Things, IoT, right? So many things are happening in the world. We will have data in the order of 10 raised to the power 30. It's huge. The word is geopower. I think you may not have heard this term, but this picture is also a bit old. So this is new vocabulary, right? Megabyte, gigabyte, terabyte, petabyte, exabyte, zettabyte, yottabyte, rondobyte. By the way, in Hindi also there is some, for size there are something, right? So I think, but we normally represent in these byte. Byte means 8 bits, right? So this is the new terminology. Again, this is another estimate. If a human takes 10 photos per day on an average, total photos clicked in the lifespan will be this, which requires 1 terabyte. Of course, assuming reasonable size of the photograph, right? These are some of the things, so yottabyte, although these things are, whenever we made, this we made few years back, but now this is old actually. We are trying to estimate something, and every time, things are old. Now, important problem. There are 2 issues with data storage. One is size. The amount, you require a lot of space. See, terabyte you can store in this hard disk, petabyte in this kind of pod. Exabyte, you require 2,000 cabinets and fill 4 storage data center. That makes up a city block, say in New York. Zettabyte, 10 raised to the power 21 will fill 1,000 data centers, about 20% of Manhattan in New York. And yottabyte will fill the states of Delaware and Rhode Island with millions of data centers. You see the size, 30 kitni chahiye, right? To store data. And this is very costly. Of course, this estimate is a bit old in 2008, but for comparison, it is sufficient to know. US GDP was $14 trillion in 2008. European Union was $18 trillion. World GDP was $61 trillion. Or storing 1 yottabyte at that point of time was $100 trillion cost. So cost is also too much. Space is also too much. So we have to look for the solution. You have to look in nature, biology, right? That is the only place. So I'll just show you what are the possibilities. So what is the ultimate storage device? So you need to look at, I mean, this is a quotation which is given by Edelman. Edelman, you might be knowing, if you have done a course on cryptography, Edelman was RSA crypto system, one of the person. So biology and computer science, life and computation are related. I am confident at their interface, great discoveries await those who seek them, right? So this is given by him in 1998. So I think here I missed. Okay. So basically, there is an area known as biomolecular computing involving peptide computing, protein computing, membrane computing. This is all theoretical. RNA computing, DNA computing, bacterial computing is very practical. Okay. These three areas are very practical, out of which, if you focus on DNA computing, it is, doing computation with a bunch of DNA strands. And the problem of DNA storage requires generating some sort of DNA strand. So, it is somehow related actually. And there are different applications, DNA ties, DNA nanostructures, strand displacement. All these things have been used actually for storage, DNA cryptography, and especially DNA storage system. What is the capacity? 1 gram of DNA can store 455 exabytes of data. Density you see. So 2 gram of DNA can store entire internet. 1 kilogram of DNA can store anything ever produced on this planet, right? Only 1 kg DNA. So that is a remarkable density, right? Right? But there are certain challenges. But this is what is motivating everyone to actually get this thing done pretty quickly, right? So this is the. So what is DNA actually? The DNA, you can have this phosphate backbone and these base pairs A and T and C and G, which are complementary. So there are 3 hydrogen bonds between C and G, and there are 2 hydrogen bonds between A and T. You can see adenine, cytosine, guanine, thymine. I think some of you might have read some biology at some point, I hope. Okay, but okay, so this process is known as single strand DNA forms hybridization with a complementary base pairs, right? So there is a helical structure that is formed. This is a very common thing. Now, 2 machines which are very important, and actually one of them is really bottleneck right now. So this is a pen drive level machine for reading DNA, already there in the market. So you can read DNA. If some DNA strands are there, you can read it using this machine. But in terms of, so this is for reading, okay, and the other one is for writing. When you want to write DNA, okay, synthesis of DNA. So reading means sequencing, and because of genomics projects, a lot of work has been done there, and people have, how to read data. You want to look at the DNA of cockroach, you want to look at DNA of cow, different plants, and sequence them. So a lot more progress has happened there in that area. But synthesis, there are challenges. Okay, it's very, not much progress has been done in terms of technology, but there are companies which are making different kind of machines there. By the way, a rough comparison in terms of access time, flash, hard disk, tape, DNA-based archival, a couple of hours it takes to write. By the way, durability is centuries, right? So all your data that you store in hard disk or any other medium will decay after 40 years, 30 years. But here you can store thousands of years, So another beautiful thing to do, right? And you can see that world's whole data can be stored in 1 kilogram of DNA. This is something as I mentioned to you. Now there are two approaches. Earlier approach was sequence based. You basically, any doc file, PDF file, or JPG or whatever is nothing but byte strings. So now these byte strings you require somehow convert into the DNA strings, some mapping. Okay, you convert them and then you reverse them back, right? This method is known as sequence based approach. So that is one. And here they are using next generation sequencing. I'll talk about it later on. Another one is structure based. So you write data on the structure of DNA. So like that double helix, you make holes. Wherever there is a hole, you call it 1. There is no hole, you call it 0. And that is how on the, in the structure you can store data. Density, you will reduce it up to 30%. So here 1 gram can store 455 exabytes. There it will, we don't have that much flexibility. It will reduce by 30%. Okay. But still, there are certain benefits in that technology. So both of these two things are emerging. But here it is very fast rewritable. The sequence-based approach, rewritable is a difficult task. So these are two technologies. I'll discuss these things later on. Now, sequence-based, there are these 6 steps that are required. Encoding of data into ACGT, using some synthesizer machine to synthesize the DNA strings, store it in the test tube. You can store it on the glass. You can store it on a paper if you want, right? Or you put it in your garden, the test tube. It doesn't require electricity, right? See the flexibility you have. And then retrieval, and then there is a sequencing. You will read by some sequencer machine and then convert it back. So basically a software is required which will do the encoding and decoding, right, of the data. So in between synthesizer, there is a machine which will do synthesizer, means DNA writing, and sequencing means reading. Okay, these two things are. So there are, in the course, we are going to look at each of these steps. What are the challenges? What are the progress to what we have done things? Companies such as Swiss Biosciences, Catalog DNA, they are developing DNA printers now. So it requires expertise in VLSI, semiconductors, mathematics, computer science, physics, biology, chemistry. Right, so they have everything. Okay, so it's something really interesting. And of course, you can ask what happens if there is an error. And that's where I come in. So basically, we need to design codec which can detect and correct errors. The software will take care of all this error correction. Okay, so different kind of things. We spend some portion. So actually, we are going to spend some portion, the course, certain portions we'll cover from these different techniques. Now, brief history of DNA storage. In 1960, Robert Wiener and Neyman actually, they gave the idea of genetic memory. And in this book in 1986, Richard Dawkins, I think you must have read this book, Selfish Gene. I don't know whether you read it. I think you don't read these days, right? I mean, that is a problem. But so I had that copy of Selfish Gene earlier. So here actually, he actually mentioned about DNA storage is as a possibility. Okay, yes. Sequence, the other way is structure based. You store on the structure of DNA. So basically, okay, I will show you. Here, you see, that is a DNA one side, right? So basically, you put a hole using an enzyme known as restriction enzyme, which can, put a hole on the DNA, phosphate backbone. Okay, so if there is a hole, means there is a 1. If there is no hole, it's 0. No, not on ACGT, on the phosphate backbone. ACGT is a base pair. There is a phosphate backbone. I think if you look at the DNA structure here, let me show you. You see, in this DNA, we are putting holes here. Okay. This side and this side, both. Now, you may say that, I mean, there should be a framework, right? So, of course, distance between two holes is around 25 base pair. So that, there is no confusion, and then you should know the position. So there is a lot of work that was done in 2020. It's very recent work, by the way. Okay. So where they have done this successfully. Okay. But there is a enzyme, TFLGO, which can do rewriting. It can erase also. The sequence-based approach cannot erase data. Here you can erase data also. You just work as a actual natural hard disk. So that kind of benefit is there. All is very new, so it is still evolving, right? Then the first demo of the concept of linear storage was in 1996 when Joe Davis actually encoded a 35-bit image into DNA. Okay, that was the first demonstration, 1996. And here is a early timeline for DNA storage. And after that, you see 1999, some work was done. But these are all very, very small things, 1999, 2001. And all these papers, they stored on some living thing actually, some cell or something, transform everything in the cell and so on. So all kind of things. Actual thing happened by this guy. And I mean, he's my hero because George Church is a very famous person at Harvard. You should know that he is a professor, of course. And each of his paper creates one new area most of the time. He's having 50 companies in his portfolio, right? So very, very powerful person. So I hope, so you see every time he does something interesting. For example, in the DNA synthesis, he developed another new method. Okay. So even in the DNA storage, he's still improving the techniques, what he did it first time. So this Church group actually in 2012 did the first, work where they demonstrated by storing data into the DNA and retrieved it successfully. Okay. But error correction was done manually. There was no algorithm that was used in his paper. Immediately, and almost the same time, Nick Goldman, who was working with Emily, she is now a founder of CEO of Twist Biosciences. I think that company is doing remarkably good. Actually, one of the pioneer in DNA storage technology right now, Twist Biosciences in Silicon Valley. So they actually collaborated with Microsoft Research in 2016-17 actually, after the first paper by Microsoft Group in 2016, this paper here. Okay, so basically, this was the remarkable work. Goldman and Emil, and then some other people, they did another work, the Goldman et al. Paper. That's very important. And we actually modified Goldman paper in this software DNA cloud. Okay, so you can look at the Wikipedia page where the entry is given, how this thing happened. And then after that, so we have written some more papers, like this is from our group. This is Olgika, another person in UIUC. Robert Grass in Europe, he has done some, he's still doing a lot of interesting things. So this is how basically, very, very early papers, okay? And these are the important ones. So George Church, Goldman, Emil, and Terence Dess at Microsoft. So Microsoft has around 35 papers till now from 2016. Okay, and some of them are really very, very beautiful. And they have very strong group of DNA storage. So if somebody wants to go and get work there as an internship, that is a possibility actually, anyway, in Seattle. So these are some early papers. As I mentioned, there is 2012 paper, 2013 paper by Goldman. And then immediately, we did the work in 2013, but we published it in 2014, that software, DNA Cloud by Sharin. And in 15 also, we modified some further work. So this was the turning point when Microsoft did this work. So they got interested and they joined hand with Twist Biosciences. And finally in 2020, they formed this alliance, international alliance. We are also a member of there. So to develop the standards for DNA storage technology. So that is what is important paper. Do we have, how much time we have? 8 minutes? Okay. So I can talk briefly about our work for two of the papers, 2013 and 2017, because of the historical things I am taking up. So this is the church paper model. So we have data source. He uses some run length encoding, DNA coding. Then of course, these two machines of reading and writing, coding. And the chunk architecture was index bits and information bits. Total length was 115, number of chunks were this. They stored 5.27 MB book actually. And there was no error correction in their technique. Okay, this is the original paper by Church. Then this is the Goldman paper. So they modified, they used some sort of 4-fold redundancy. The chunk length was 117 there. That was again a paper in 2013. Okay, so these two, you can see that Huffman coding is used for data compression there, right? Those kind of things are there, right? So this actually paper, when we realize that the chunk architecture is fixed, it is creating a problem. So we need to vary it. And that was done by Shalin Shah. And Dikshita was my PhD student. So this tool is still available, you can use it. Storing big data on DNA. So I'll give you a brief example rather than going into details. But we were able to store 115 exabytes per gram. In their previous work, it was petabytes. We were able to reach exabytes actually, theoretically, right? We have not done the experiments, but it is costly, right? And doing experiments in India is anyway very difficult thing. But anyway, here is a code book that we use, chunk architecture. So I'll give you an example, see, this is a Goldman work, binary file, the byte string converts into base 3 encoding, 012 string, and then converted into ACGT using this kind of map. And then they use DNA fragments like this for 4-fold redundancy. So very naive error correction technique that he used for correcting the errors. So we replaced here using our coder, and here is our, let me show you an example, I think that will be better. So DAICT, ST values are this, right? Corresponding ternary from that code book that we created, you can get these ternary strings, okay? Then we have, separator, different separators, file extension, it's a text file or what, txt, and then size. And we paired it with 0, so that it is multiple of 99. We want chunks of size 99, okay? So we paired it with zeros. So here is the final string. Then this string is converted into ACGT using this map. We have done a lot of work in this map later on, but this is something, this map ensures one thing: whenever you convert any ternary string into ACGT, no ACGT is repeated anywhere. Like AA, CC, GG, TT will never come because of that map. Let me show you how. Start with 0. You can assume that previously written nucleotide is anything. So suppose we start with 0. Suppose previously written nucleotide is A. So 0 will go into C. You're right. Then go to the next one is 2. 2 previously written nucleotide is C. So 2C will give you A. So here it becomes A. Right? Like that, you can keep on continue. This is the conversion which can convert ternary string into ACGT. Then we have this final string, then there are 2 chunks. We can add chunk architecture. So we have added chunk kind of chunk 1, chunk 2, index for both the chunks. Okay, so this is how DICT is encoded like this, 2 chunks here. Yes. Yeah, so there are certain efficiency issues. People have done the binary things also. Ternary is interesting. So we will discuss more about it, why ternary, okay? And why not quaternary? Quaternary could be taken directly. You will see, we'll talk about 2 kind of optimal parameters like information density, how much information you can pack in the DNA, right? So normally, see, if you don't do any error correction, 2 bits can map into A, 2 bits can map into C. So 0001, 1011 can map to ACGT, right? No error correction. This is the best possible. But as soon as you introduce error correction, it reduces. So the idea is, how do we construct something which is close to 2? 1.88, 1.99, 1.7771. There are different schemes. I'll talk about it later on, right? Yeah, so there are many constraints. We'll talk about it. There are many constraints. I think around 10, 15 constraints are there. So you can solve it mathematically. You can, although in the industry they do experiments and simulations and they say that, okay, millions of times that we have run the things and we are safe. We'll talk about it later on. Okay. So we modified another version of DNA Cloud. This time Vijay Dhamilia. I think you must be knowing Vijay is now a sadhu in ISKCON. So after this work, he got a PhD at Iowa State University, but he left that, and he's in Ahmedabad. You can meet him, ISKCON. Okay. So that also happens after doing this kind of work. So here is a comparison, actually, of the Goldman result, and you can see that the amount of nucleotides ACGT that we use is very less as compared to Goldman, our method. So I think probably I'll stop here. So next time I'm going to talk about bacterial storage and a couple of other things, right? So I hope Thursday is a class, right? Yeah, so Thursday we'll talk about it, right? Thank you.